vectors and the host organisms. So, another, what do you mean by restriction enzyme? Explain with an example. Restriction enzyme cuts the DNA at two ends of the specific gene and a restriction fragment is obtained. Restriction enzymes belong to a larger class of enzymes called nucleases. There are two kinds: extro exonucleases and endonucleases. Exonucleases remove nucleotides from the ends of the DNA, whereas endonucleases make cuts at specific positions with the DNA. For example, a restriction enzyme called LUE cuts DNA everywhere that its target sequence AGCT occurs, but nowhere else. There are many different restriction enzymes which each of which recognize and cuts its own unique target sequence of DNA. Restriction enzymes were discovered in bacteria in the late 1960s. Nature. These enzymes protect bacteria against foreign antigens. Next question we have: what is gel electrophoresis? The cutting of DNA by restriction endonucleases results in the fragments of DNA. These fragments can be separated by a technique known as gel electrophoresis according to their molecular size. By using restriction enzymes and gel electrophoresis is together, we can examine differences in DNA sequences. For example, the restriction enzyme cuts the normal hemoglobin allele into two pieces, but it cannot cut the sickle cell allele, providing a simple test for the disease allele. With the diagram, explain the steps involved in recombinant DNA technology. For some time, we can skip this question. No, read it. Sequences of steps in recombinant DNA technology. Specific restriction enzyme is selected. Cell culture with required gene in the cells is obtained. Restriction enzymes cuts the DNA at two ends of the specific gene and a restriction fragment is obtained. Same restriction enzyme cuts a matching DNA sequence from a plasmid. Okay. Ligase joins the restriction fragment in the place vacated by the cut DNA segment of the plastid. The plasmid becomes a recombinant plasmid containing a foreign DNA fragment. Its DNA is the recombination DNA. Since plasmids can carry foreign DNA, they are called clonal vectors. Bacteriophages can also function as clonal vectors. The recombinant plasmids then enter bacteria. Then bacteria divide into recombinant plasmids, replicate along with the bacterial DNA. A large population of bacteria, more than 1 million, containing recombinant DNA can be obtained in less than 10 hours. Then we have multiple identical copies of DNA fragments inserted into plasmids or bacteriophage. Bacterial virus then obtained and preserved in a DNA library. These DNA fragments are the cloned DNA. Then we have next question that can you suggest a method to remove oil hydrocarbon from seeds based on your understanding of our DNA technology and chemistry of oil? So recombinant DNA technology, rDNA, is a technique used for manipulating the genetic material of an organism to obtain the desired result. For example, this technology is used for removing oil from seeds. The constituents of oil are glycerol and fatty acids. Using rDNA, one can obtain oil-less seeds by preventing the synthesis of either glycerol or fatty acids. This is done by removing the specific gene responsible for the synthesis. Describe the steps involved in PCR technology. Polymerase chain reaction, which means PCR as a short form, is to make a huge number of copies of a gene. In this reaction, multiple copies of gene or DNA of interest is synthesized in vitro using sets of primers, small chemically synthesized oligonucleotides that are complementary to regions of DNA, and the enzyme DNA polymerase. Generally, there are three steps in PCR repeated by 30 or 40 cycles. We have first step, denaturation at 94 degrees Celsius. The double strand DNA melts by breaking hydrogen bonds. Single strand DNA short chains are formed. Okay, step 2, annealing at 54 degrees Celsius. Ionic bonds are constantly formed and broken between the single and single stranded primer and the single stranded template. More stable bonds last a little bit longer and form template plus primer of dsDNA. The polymerase attaches and starts copying the template. Step 3, extension at 70 degrees Celsius. The primers that have a stronger ionic attraction to the template are with few bases built in. The bases are coupled to the primer on the 3 adds DNTPs from 5 to 3, reading the template from 3 to 5 side. The cycle then repeats itself because the process uses a special heat resistant form of DNA polymerase. It's not necessary to add new enzymes after each heating stage. Now we have another question. How is insertion of recombinant DNA done in host cell organism? How is insertion of recombinant DNA done in host cell organisms? Here we have the answer. There are several methods of introducing the lighted DNA to recipient cells. Recipient cells, after making them competent to receive, take up DNA present in its surroundings. So, if a recombinant DNA bearing gene for resistance to an antibiotic is transferred to E. coli cells, the host cells become transformed into ampicillin resistant. If we spread the transformed cells on agar plates containing ampicillin, only transformants will grow. Untransformed recipient cells will die. Okay. Then we have the ampicillin resistance gene is this in this case is called a selectable marker. Define transformation with examples. Transformation is procedure through which piece of DNA is introduced in a host bacterium. Normally, the genes encoding resistance to antibiotics such as ampicillin, uh, chlorophenicosol, tetracycline, or uh, kanamycin, etc., are considered useful selectable markers for E. coli. The normal E. coli cells do not carry resistance against any of the antibiotics. Okay. Now we have another question that give the applications. No, we have what are transgenic bacteria illustrated using one example at least. So we have transgenic bacteria contain foreign gene that is intentionally introduced into its genome. They are manipulated to express desirable gene for the production of various commercially important products. Okay. As example of transgenic bacteria is E. coli. In the plasmid of E. coli, the two DNA sequences responding to A and B chain of human insulin are inserted so as to produce the respective human insulin genes. Hence, after the insertion of insulin gene to bacterium, it becomes transgenic and starts producing chains of human insulin. Later on, these chains are extracted from E. coli and combined to form insulin. So we'll do some more questions. That we have another question. Give the applications of recombinant DNA technology. Okay. So application and uses of recombinant DNA technology. We have some that genetic engineering recombinant DNA technology has enabled the understanding of structure of eukaryotes genes and their components. Second, we have genetically engineered bacteria are employed to synthesize certain vital life-saving drugs, hormones, and antibiotics. Example: antiviral, anti-cancer, interferons, human growth hormone, and somatostatin, etc. Through recombinant DNA technology, the genotype of plants are altered. New transgenic plants which are resistant to diseases and pest attack have been produced. I have finished this whole page. Move to the next page. Genetic detects defects in animals as well as human could be corrected through gene therapy. Genetically engineered bacteria are called superbugs. Superbugs can be degrade several uh, aromatic hydrocarbons at the same time. They are employed in clearing oil spills in the ocean. Now we have today's last question: how genetic engineering helps in producing pest-resistant crops? Some of the applications of biotechnology in agriculture are the production of pest-resistant plants, which could decrease the amount of pesticide used. Mm, toxin is Bt toxin is produced by bacterium called Bacillus thuringiensis, Bt for short. Okay, Bt toxin gene has been cloned from the bacteria and been expressed in plants to provide resistance to insects without the need for insecticides. In fact, in effect, created a biopesticide. Examples are Bt cotton, Bt corn, rice, tomato, potato, soybean, etc. Post resistant plants. Several nematodes parasite a wide variety of plants. A nematide, Miliodigene incognita, incognitia, infects the roots of tobacco plants and causes a great reduction in yield. By using the RNA interferase, RNAi involves silencing of a specific mRNA of the nematode. The consequence was that the parasitic could not survive in a transgenic host expressing specific interfering RNA. Transgenic plant therefore got itself produced uh, from the parasite. So this was our last question for today, and in another video we'll discuss some more questions. Until then.